Is the microphone working? Oh, no, it's working. Okay, so I would like to welcome everyone to this short presentation, and I'm in an awkward position, but I will start with the beginnings. So the idea that we can shape behavior by awards and punishment is very basic. I mean, it's natural. For as long as people cared to write down ideas, they knew it. And I here have a citation from Aristotle just to make the point. But why I'm in an awkward position? Because yesterday you had the unique opportunity to listen to a guy who was starting with more or less the same slide, and Pavlov. Because Wolfram Schultz is probably the most prominent, or eminent, I would guess, researcher in this area, and you had all the introduction I have prepared today, but in the better version. Still, I will try to do my job. And luckily for me today, the previous speaker also did an excellent introduction to the dopamine system, but I will start at the very beginning. I mean Pavlov. And why is Pavlov so important? Because for the first time, someone tried to investigate whether a simple reaction can be conditioned. And this is the very beginning of conditioned behaviors and behavioral studies. And from these behavioral studies, from their development, starts neuroscience. The first 30 years were spent on conditioning, on instrumental reflexes, etc., etc. But for all this time, there were theories on how this works on the brain level, yet there was not really a way to test this hypothesis. So the breakthrough was the Olsen-Milner experiment. It was a very basic idea. As soon as people have perfected electrodes and learned how to stimulate the brain, they started placing the electrodes, well, not really randomly, but more or less all over the brain and monitoring for reactions. So Olsen-Milner observed that if they put an animal into an open field, and in one area of this field they would stimulate the lateral septum of the brain, the animal would really like to stay in that area of the arena. And this is essentially the same as people do today with optogenetics. But they were first with simple stimulation. Then they extended this experiment with this one that you probably know, where they have introduced in a Skinner box by Bruce Skinner, who is here. And the rat had the option to press a lever, and this would cause a stimulation of the lateral septum. And as they observed, rats really, really liked to do that. I mean, they would do that constantly. So whatever action, like this operant action, pressing the lever, was paired with the stimulation of this part of the brain, was reinforced. I mean, it would become much more likely to happen again if it was followed by this stimulation. And so the research on the reward system of the brain started. People learned that there is a specific place in the brain that if you stimulate it, it will reinforce the action that led to this stimulation. And this really exploded then. So the most important thing, I already tried to make the point on the previous slide, showing that people have studied all sorts of animals and humans in behaviorism. But all these experiments on stimulation of the brain were of course done in humans too. Here I have an example which is a really, well, horrible one, but it's an actual study where you can see on a Röntgen coronal image of the human brain. What you can see here is the human striatum, and this black line is an electrode placed all the way down to the lateral septum. And as the authors noted, that if a human was stimulated in this area, they would like it really, really a lot. And even they used the same language in describing the reactions of the patient to this stimulation, had to be disconnected, as Olsen-Milner has used when describing the rat, had to be unhooked. So this system is highly evolutionarily conserved. At least it is in mammals, but definitely it works the same in birds. I have no idea about other vertebrates. Okay, so the next breakthrough came from two sources. First of them was Carlson, who has discovered that dopamine was not only a metabolite, but it was also a neurotransmitter. And at the same time, pharmacology started, well, this was done because he was interested on how drugs work. In the very beginning, people have been developing drugs. They knew they worked to treat psychosis or maybe depression, but they had no idea why. So his research was to investigate how antipsychotics worked, and this then actually went into developing the selective antidepressants. But at the same time, there was much discussion, what does dopamine do? And the first good answers were from Roy Weiss, who have shown that all these reactions in the brain that were in the reward system, they were mostly dependent on dopamine. At that time, people were wondering what this was, journaling, what was dopamine. There was some confusion. Roy Weiss said, no, it's dopamine. And also, he said one very important thing, it's in his paper from '78, that dopamine plays a critical role in the subjective pleasure. And this sentence had such a big influence, I mean, beyond science. This is the whole source of dopamine as happiness. Of course, as if you listen to yesterday's lecture, it is not really the case. You heard it from Wolfram Schultz himself that dopamine is something completely different. And also, dopamine has many functions. It's not only a signal in the reward system. It controls lactation, controls movement. It has many functions. So, but still, this lives until today. I have a slide which I'm not showing today, which discusses the dopamine deprivation diet, which is very popular apparently in the United States, or the dopamine low. It's called meditation. I think that is supposed to help the brain to adapt to high stimuli. It's all based on this sentence, which is not even correct. And Roy Weiss himself admits that in modern days, though there is still some discussion. So, okay, this one you have seen before, and at least yesterday, but why is not dopamine the signal of pleasure? And I will just try to do it as a very short introduction on this. So Wolfram Schultz and colleagues have studied the activity of dopaminergic neurons, presumed dopaminergic neurons in the substantia nigra pars compacta of rhesus monkeys who were the recordings were made while they were shown visual stimuli, and these were paired with small dose of sugared water, or it was, I think, a juice. It was a very sweet juice. So monkeys really love the juice. They like it. So when they receive a few drops of this juice, there would be a reaction of the dopamine neurons. Such response would be consistent with the idea that they signal pleasure. There is a stimulus, a reward, and then there is a response. However, when every time before the juice was delivered, like 1 second before, they would receive a visual cue, then all the activation of the dopamine neuron would occur upon the stimulus, and there would be essentially none when the reward itself was delivered. So the dopamine does not encode reward itself. It encodes the information that predicts the reward, and this is a prediction error because whenever, for instance, when they cheated and they have shown the stimulus but there was no juice, there would be a pause, silence. Or if the portion of juice was larger than before, there would be still a second peak. So this is the expectation. This is the error of the expectation. When this should be high when the stimulus is detected, this should be zero if there was no error in prediction. Okay, this property of the dopamine neurons is really interesting because people speculate that they may encode a quantitative error, quantitative prediction error. Why is it so important? It's not only the dopamine neurons which have this property that they predict rewards. Many types of neurons, including noradrenergic, serotonergic, and even some of the cortical neurons, also respond to stimuli that predict rewards. But as far as I know, it is only postulated to the dopaminergic neurons that actually you can decode from the response what is the expectation and what is the level of error in the expectation. This is done also from the work of Schultz. And on the right, I have the Rescorla-Wagner equation. This is a slightly different version from what you have seen yesterday. Rescorla-Wagner have developed this equation when they were discussing the theoretical underlying mechanism in the Pavlovian conditioning. And I just wanted to point to a few things. Well, first of all, that in the Rescorla-Wagner equation, it discusses the accumulated value. So it shows how the total value of what is the total value of predicted information so far. This is different from the equations I will show you later because the expectation of value has to do with an action and the value accumulated is not exactly the same. But the equations look the same, but please keep in mind this difference. Also, in the Rescorla-Wagner, they had two parameters which modified the prediction error. One was the learning rate, and the other was the salience of the cue. Let's not discuss the salience of the cue. The learning rate will be important later, and it was the amount of information derived from this prediction error that was just experienced. Okay, so just a few more examples which actually were also shown yesterday is that the dopamine signals also correlate very nicely with the delay discounting, which is an essential property of these neurons. And well, I guess I will stop here and just say that At the time that we were starting this project, which is the research on what mechanisms control the activity of dopaminergic neurons, we wanted to test that if you manipulate this signal, these bursts of activity, how will this affect the prediction error? And this was done in collaboration with the group of Tomasz Błasiak, and this is the group from where the previous speaker is, who were able to do this elegant experiment and just show this. Okay, so here you have an example from Wolfram Schultz's research when you see the recordings of single-unit recordings which show an increase in firing of a dopaminergic, presumably dopaminergic neuron upon a stimulus. And this is a recording from the mouse, from an anesthetized mouse with a tetrode from the ventral tegmental area, where the neuron is firing spontaneously, having this type of tonic activity, which is, I don't know, it's 0.5 to 2 hertz. I don't know where Gabriela is. Okay, so it's about 0.5 to 2 hertz. And when you apply an NMDA, and an NMDA is the eponymous agonist of the NMDA receptor, you will observe this. This is not a reduction. This is actually the amplitude appears lower, but this neuron then starts firing in bursts. So it no longer fires in more or less evenly spaced episodes, but it fires in these packed short bursts with pauses. And this mode of activity, the phasic burst activity, is supposed to be very efficient in releasing dopamine. And by that I mean that even if you had the same number of spikes over a period of time in this mode compared to this mode, this should be releasing more dopamine than this. So people assume that when this happens, dopamine gets released in the striatum and things get done. Okay, so note this. So this happens when NMDA is being applied. And naturally, of course, this would be projection, glutamatergic projection from the forebrain or from the hindbrain, informing the dopamine that now is the moment that something important happens and they should promote the activity in the striatum. So what we could do is we could remove these NMDA signals. How? I assume that you have mostly background in modeling and bioinformatics. How good are you with genetically modified mice? Should I be going in depth here? Any hints? I assume that you know at least something, but we were working with conditional mutations, and by that I mean that we are able to remove one subunit of the NMDA receptor at the NR1 subunit in specific types of neurons. Why the NR1 subunit? The NMDA receptor is a tetramer which is composed of 2 NR1 subunits and 2 other subunits, usually NR2. NR2A, sorry. And without NR1, there is no NMDA receptor. It was actually shown that if you genetically remove the NR1 subunit, there are no functional NMDA receptors. Now the trick is that when you do it completely, that if you remove all the NR1 genes in all the cells, there will be no animal because it will not be born. It's little. So the idea was then to somehow limit the mutation to specific types of cells. This is being done by the Krylov-Spi system. That's the most typical approach. And here I have 2 examples of mouse strains which have the mutation restricted to either dopaminergic neurons by using a combination of a Cre recombinase expressed under the dopaminergic transporter promoter and the NR1 FLOX gene. So in the end, this component is expressed only in some cells, and the enzyme produced by this component will remove this gene, but only where it's present. So only in dopaminergic neurons. And here's an example showing that the recombinase, that the enzyme that will inactivate the gene, is only present in these neurons of the ventral midbrain, presumably dopaminergic neurons of the ventral midbrain. The second line we have been using in these experiments was kind of a, well, the other side of this signaling pathway was the D1 CRE-RT2, which is the dopamine D1 promoter driving the expression of the Cre recombinase. And in this line, the mutation would only occur in the target areas where dopamine goes. The D1 receptor is expressed by those neurons that receive dopamine projections. Not all of them. About half of them in the striatum and some of them in the cortex. But this mutant line mostly works in the nucleus accumbens of the striatum, which is shown here. Now, a second trick that we use is that these lines actually have inducible mutations. These enzymes that produce the mutation are not active until you give the animal a special compound, tamoxifen, a synthetic steroid, which will activate the enzyme. So these mutations were induced in adulthood. The animals didn't develop normally, there was no mutation, and then when they reach 8 to 10 weeks, we induce the mutation. I guess it has to be enough for today. And okay, so what happens when you do that? Just to show that this impairs plasticity, This is really, well, maybe let's start with this one, on the right. So you remember this, that if you apply an MDA on a dopaminergic neuron in the ventral midbrain, it will change activity from tonic to phasic. And this is just the whole trace. These are the fragments of the trace. Okay, and below you have the mutant. So the tonic activity would appear, at least in this specific case, lower, but it was not significant in the population, and an MDA had no effect. So we would say that those neurons lose their ability to produce these bursts of activity, though this would not be completely accurate, because there are other mechanisms, the NMDA receptors, that could trigger burst activity. But the most classic one, and the one which is usually indicated as the main one, is NMDA receptors. Okay, on the other hand, when we do the mutation in the D1 neurons, the ones which receive dopamine, what we could show is that they lose some plasticity. Is that really important? Well, it's not all of the neurons in the area. So the loss of LTP in this case was partial. But apparently the mutation works. You impair the plasticity, and those neurons, we assume, cannot produce plasticity similar to the LTP. What are the consequences then? Now, to study the effect of such mutation on behavior, on the decision-making process, we have been using this probabilistic reversal model. Are you familiar with it? Do you know what the two-armed bandit problem? You should be. You have two choices, and you don't know what are the outcomes of those choices. Those choices are probabilistic, so they may or may not give you a reward. If you try to sample those choices, you will notice that one of these options will give you more rewards than the other. So you would assume that you should be focusing on this better choice because it is more profitable. However, to make the task more complicated, the probabilities of getting a reward for one of these choices is not constant. It changes. In the human task, you can actually change it a lot. In the mouse task also, but it's a bit difficult, so we do it just once. We reverse the probabilities in the middle of the trial, and we have 60 trials in one. Combination of probabilities of reward and 60 choices in the other. In this case, the reward is really a big reward because you have an animal that had limited access to water, and they may receive water if they, have luck, and the choice they selected awards them with a bit of water in the food pot. And what you can see is basically summarized here, that they will be, this is the rolling average of choices on the left side, and you can see that this is the probability. So 1 would be previous 10 choices on the left side, 0 is previous 10 choices on the right side, and blue shows which side has the higher probability of giving the reward. So what you can see is that initially in the first few sessions the animal learns the task, but do look that the slope of this curve, how soon it will change the preference, becomes sharper and sharper. By the middle of this task, it will change very quickly and stay on target. Okay, what about the mutations? This will be really fast. We actually had another one mutation, which I guess if you remember the first day there was a lot about metabotropic receptors. We also target the metabotropic glutamate receptor. Let's not go into the exact mechanism of how this works, but just at the behavior. Now these faded lines show the control groups. These have no mutation. So this will be the normal behavior, and I'm very happy to say that the control groups, if all the experiments behave more or less the same. After a few sessions, the animals learn more or less to prefer the better side, about 80%. This is the entire session, including the change in probabilities. They reach a plateau, let's say it reaches a plateau more or less here after 10 sessions, and stay on it. The mutations, first surprise was that these mice actually did learn the task. So they were able in the end to, at the end they did acquire the preference for the higher probability of reward, the same as control mice. They were slower. These mice, they were generally they did not favor as much the higher choice of higher probability of reward, while these animals were completely unaffected here. And if you look at this task from the point of view how this process works, there are only two types of decision in the task. You can either decide to do the same choice again or do the other choice. So if you consider that, let's say that previously the choice was rewarded, then after a win, you can stay or shift. And if the choice was not rewarded, after a lose, you can stay or shift. A perfect strategy in this task is to stay after every win and shift after every lose. This will theoretically give you the best amount of rewards. The animals are not as clever, humans neither. I mean, this is not really an animal thing, but you can see that win stay, they really do. I mean, they go about 90% of win stays, and there's just a minor difference, a slightly lower level of win stays in the mutant mice, and it happens both in this strain and this strain. The loose shifts, they often are about 50%, sometimes they are higher, but usually 50%, so they don't really use this information as much as this. And yeah, in this case, what we also observed, because this difference was like, well, it's small, it's significant, but it's so small. Whatever it means, does it mean much? I will go deeper into this when we get to the modeling, but what we noticed that the difference in behavior was elsewhere. The time to commit to the decision. So the big difference was that this strain, the one with no burst activity in dopaminergic neurons, this burst activity that is supposed to produce efficient dopamine release, these are the control mice, and they would usually make the decision 5 seconds from the moment that they receive the cue that they can make a choice. But the mutant mice were much slower throughout the session. This is an average of 15 sessions, 120 trials, time to decision. There was no limit on the time of the decision. If we put a limit, those mice would fail a lot because they would exceed the time. The same was the case in the second strain that was slightly impaired in the percent of better choices, and there was no effect in the strain that had no impairments observed. So this appeared to be a much larger effect than the change in win stay. And in general, we noticed that what these mutations impaired was not the ability to choose, but the efficiency of choosing. And it would make sense, because probably, even if these animals had no burst activity of dopaminergic neurons, probably other compensatory mechanisms could play a role, or maybe there were some additional circuits that could encode this information, but clearly, this one is most efficient. Okay, so let's get to the modeling. I think that you might know more about the work of Sutton and Barton than me, and if not, you probably haven't had an introduction yesterday during the lecture from Schultz. But in general, let's consider this problem that the animal, the agent, may perform an action, which is the choice of the left or the right nose poke, one of the two operands available, which will then result in an outcome, a reward or lack of reward, and a change of state. Well, in this case, there is no change of state. We assume that the animal is in the same state during every trial. So the environment is constant. We don't have to consider that the animal choosing one of the options will limit the possible options in the further steps. The option is always the same, so we don't consider the state. We only have the action and the reward. And the simplest way to model the animal behavior would be to say that let's build a model where we have the animal having some expectations. It will have an expectation of reward of choosing A and B, and this is defined as Q, A, N. So it's the expected value of action A at step N, considering the state which is constant, so I don't put the S as an additional parameter. This is the softmax policy, and the probability of selecting A or B is based on the difference in the expected values of the options and this beta parameter. The beta is called inverse temperature, and this parameter is essentially saying how random is the behavior. When the parameter is 0, then the probability of selecting A or B is equal, and it's not dependent on expected value. So at 0, the behavior is completely random. At infinity or at high values, only the option with higher expected value will be selected. Okay, so the animal chooses, there is an outcome. It's a reward or lack of reward. So then is an update of an expected value, and the update is essentially the previous expected value plus the prediction error. If there was no error in prediction, and there's almost always a small error in this algorithm, then there would be no change. But this value is almost never 0, and this value is modified by the learning rate. What is the learning rate? It's not the ability to learn. It's just how important is the last choice compared to previous choices. If this parameter would be 1, then all the information that the animal has is taken from the last choice. So if this would be 1, only the last choice matters, because then you will have this Rn minus Kn, then the expected value would be R. Can you see it? Okay, if this parameter is 0, the animal never learns. But if the parameter is small, then the animal will actually average the information over several previous trials. So this is not really the efficiency of learning, just kind of like a strategy. How much importance from the more recent versus longer history of choices. And okay, next choice is just using the updated information from this step. And this is essentially Q-learning, though if you look at the paper from Diane Watkins, they also had this gamma parameter and the maximum possible reward, well not maximum, and they also included a additional part of the equation which would predict lost opportunity if the specific action was taken, but we will not be using this here. Okay, so this equation is too simple because we want to predict animal behavior. And if you look at this, they clearly use more information from the trials where they had received the reward compared to trials where they did not receive the reward. So the first modification that we can do is that we introduce separate learning rates for the negative outcomes for lack of reward and positive outcomes for when the reward is present. And would expect this one to be larger than this one and had more influence on the expected value. Now, the big modification, and that I will explain in a moment why it's so important, was to introduce fictitious update. This is derived from studies on regret in humans. And the example given in those studies is the Dutch lottery, where they have, I have never experienced that, but apparently they have a lottery where they select a postal code. And the Netherlands, I think they have postal codes which can go right down to the specific house or home. And if you bought a ticket, then you win if your postal code is selected. So people buy these tickets because they're afraid that they would win and would not collect the winnings. And that's fictitious update because, of course, they don't win. I mean, this is lottery. But people are afraid of the situation that would happen, that they would win but will not be able to collect because they have no ticket. So fictitious update is kind of like updating for the thing that never happened. And in this case, this is updating for the other option. If we received a reward from our choice, then the fictitious update would be to lower the expectation towards the other choice. Because this one was good, this other one had to be bad, and vice versa. In case we have not received a reward, then we have this regret. The other must have been better, we should increase the expected value to the other choice. So this is actually a big change, and of course we've extended this by adding separate learning rates and then applied these models to see if they're able to predict the animal behavior we observed. How much time do I have? 15 minutes. Good. Okay, so I'll cut it short and say, so the fictitious models are far better at predicting animal behavior than the classic Q-learning. It's just what they are. And if you apply them to the experiments I have shown a moment ago, what we saw is that the mutations did not affect the learning rates. This is modeling, and these are the parameters, group parameters done by Wu Yong An and Rafał Bogacz, and this was using Bayesian models and hyperparameters. I'm sorry that the notation that was in the paper is slightly different, but it's the same as alpha plus and alpha minus. And what we observe is that while the learning rates were essentially the same in controls and mutants, none of these differences were credibly different, but the beta parameter, the one which controls how random the choices are, was decreased in mutants. Decreased means more, too far. Decreased means more randomness or more exploration. Is the same. Randomness and exploration in this case is the same, and we cannot tell if it's a deliberate choice to be more explorative or it's simply the loss of ability to be more focused on what is important. So a quick conclusion from the first part is that when we did these mutations, the first surprise was that the animals learned quite okay. What they did change, what did change was that they were less efficient at performing the tasks. They were slower, but not less able. And then another thing was that when we tried to predict the behavior using reinforcement models, the fictitious update, so having like not really a model-free approach, the fictitious update kind of breaks the model. You know the difference between model-free and model-based approaches in decision processes. Should I explain that shortly? The model-free approach says that we have no idea how the environment works. We just observe the effect of the choice, and the only information we extract is the effect of our choice. The model-based approach assumes that we build a model of how the environment is constructed and what are the exact possible outcomes, and we kind of more try to build predictions for longer numbers of choices. But we assume that in this case, that animals, especially in such a task, would rather use a model-free approach. Humans do it all the time, actually. But the fictitious model kind of breaks that, because it assumes that we think of the choice not performed. So this is kind of model-based, and it appears the best. Okay, the second part of my talk, and it will be short, I promise, is about building a better model of reinforcement learning. Because in the task I've shown you, the animal is in a horrible situation. They have limited access to water. They are very thirsty. They are placed in a cage where they are alone. There's plenty of light. It's not a home cage. And they have to perform a choice every 30 seconds. And that's not really like a scenario which you could compare to normal decision making. In case of humans, most of the studies are being done when someone is introduced into a magnetic resonance apparatus. They are being somehow restrained in it. I mean, it's a tube. And they also have to look at the screen, and they have 10 seconds. Fix your gaze here, 5 seconds, decision. Fix your gaze here, and you do it 100 times. That's not really a natural decision-making process. So we wanted to have a scenario where we would have a task, a probabilistic reversal task, that would not be so constrained, that would leave the animal some freedom of what they want to do. And here it comes the IntelliCages. So the idea was like this. There is this big cage. It fits up to 14 animals. And while the previous study was done only in males, this study is done only in females. Because 14 females will not fight that much. And in this case, the mice stay in this cage for weeks, and the experimenter only checks on them once daily, and sometimes even only has a look and just goes out because there is no need to do anything. The cage has free access to food, but the water is placed in 4 corners, not water or other liquid, that's important, and these water bottles, the nozzles, are accessible through these small doors. These are guillotine doors that can be closed or opened by the computer. So what we can do, we can place, let's say, like here, in 2 corners we would place water, and then in 2 other corners we would place some reward, like let's say saccharin solution, which is sweet and mice like it. And when the mouse enters this corner, these corners are small, and in theory only one mouse can fit in a corner. So this is a mouse trying to poke its nose and touch the nozzle, I assume. We cannot see it here. And you can see the cues lit up. And yeah, so the mouse can enter. It has a RFID chip here on the back. And the corner will detect the mouse. And this also has temperature sensor to confirm that the mouse is inside. And if it approaches this gate, it has a photocell. So it will open when it's detected, if we program it this way, and we receive the reward. But to make the task what we want, we gave them 100% chance to get water in 2 corners and probabilistic access to rewards in the 2 other corners. Of course, the procedure is a bit longer because it requires several weeks of training just to remove corner preferences and teach them that the positions of the reward change. And then we do the main experimental phase and alternate probabilities between 2 corners. And actually, in this case, we have used different, the same, low, high, or the combination of probabilities. And we also played a bit with rewards. We have used alcohol, we have used saccharin, and a combination of alcohol of saccharin, as we were then influenced by ideas that drugs and natural rewards have some different mechanism of learning, which is still a kind of popular idea. We also included one group where we put water as a reward. So we wanted to have a negative control. How should the behavior look like when there is actually no reward? And how the models would work if there is nothing to model? Because, I mean, models are sometimes surprisingly good at modeling something that should not be there. So, first of all, the important thing. So in this paradigm, the animals did prefer the reward. When given alcohol and water or saccharin water, they would prefer alcohol slightly. About the median was 60-something%. And if there was a saccharin solution or sweetened alcohol, they would have like 70 to 80% median preference for the reward. And in this case, I wanted to know that the access to water is 100%, while access to the reward was probabilistic in those cases. And also, we wanted to see how many attempts to access the reward they would make. And what mattered that is, if they entered a corner where the reward was present, they had to wait 2 seconds before the doors would open. Because we didn't want them to just enter randomly and see what's in the corners. We wanted them to enter, wait 2 seconds, and then the lottery is made and they receive or not the access. So they made thousands of these kind of choices where they waited 2 seconds for the outcome of of what will happen after they entered. Okay, so to the point. What we saw is that, okay, they did favor the high probability of reward for the saccharin solutions. For saccharin solution or for the alcohol and saccharin, both. It was not such a high preference in the case of the Skinner box. It was not 80%, it was 50-something, 60%. And not all of the animals, some of them actually did not. There was not much of a preference in the case of alcohol and water, though I admit both pass significance, but the effect is minor. These two are the big ones. So at least saccharin certainly works in a way that they will prefer the higher probability of reward. There was one strange thing. When we tried to look if they do the win, stay, lose, shift behavior, it seemed like they did not care. And when we started to look more in detail, it turned out that in this task, they have as much time to perform any choice as they want. So they can go into a corner, choose, and then wait 2 hours and choose again. And then do it over 10 times in 1 minute intervals. So they're completely free to do it whenever they want. They have 4 weeks to complete this task. So we thought, okay, maybe we had to look at the intervals between choices, and so indeed the case. When the choices were done in short intervals, they were very likely to change, to shift. When the choices were done at longer intervals, they were likely to stay. And it didn't matter if it was a lose or a shift. The longer time elapsed from previous choice, the more likely they were to stay, to repeat the same choice. Why? It's a matter of discussion for later. Now, the other strange thing was that this was the case when there was a reward present. When there was water, there was no such thing. When we gave them no reward, there was no effect of the interval of between choices on the shift or stay choice. And just for comparison, how it looked in the Skinner box, it was completely different. Now, this is the same, but done more elegantly with logistic regression. So this is a function which shows the probability of doing a stay or shift as a function of time on a logarithmic scale. What you see, these dots are, the entire graph is one animal. These are all the choices it made after a win, after it was awarded a reward in a corner. These are the cases when it returned to the same corner. There are just many dots and there is a big well, most of them are in this area, and this is what happens in the intervals when it shifted. And this is after a win, after a lose. And I guess you can see, especially on this right side, that when they lose and the interval was short, they would do a shift. And then they lose and the interval of long, they would do a stay. And as a result, you can see that the probability of staying as a function of time has this nice slope. And as I said before, this was not the case for water. So this is not just exploration. This is something reward related. Okay, this is a summary for all animals, and only significant effects are shown. So not all of the animals which were in the experiments are shown. The total animals is about 100, and it's only significantly affected. So most animals had a preference for one corner. We don't care about that. Almost all animals had an effect of reward. So they were more likely to select the same corner if it was previously rewarded. It was not a big effect, but it was there. And there was generally an effect, with one exception, that the longer interval, the more likely they were to stay. Okay, so reinforcement learning models. This is a bit more complex. So you remember the models I've shown you before? We said that there was these basic models, there could be modifications with dual learning rates, and then there could be the fictitious models. So we did the same. These models are on the left, and in this case, the models are evaluated by the Akaike information criterion. Do you know how it works? Okay, so you can, for a single animal and its choices, you can see how well a prediction model approximates the choices by measuring this information criterion. The smaller the number, the better. Here is the equation just in case. Now, it works only within one animal, so you can compare how different models, how well they predict the behavior of a specific animal. So the dotted line you have here is the value of the criterion for the basic model, for the basic Q-learning. If a model scores above, it means that it is inferior. Yeah, it is inferior to the basic one. If it scores below, it is superior. Okay, so what you can see is that we tested many models, including some more exotic ones, which I will not be discussing, like the noisy wind stay, which is a model which assumes no reinforcement, it's just based on previous choice, and the so-called forgetful or attention models, which have additional parameters. But we found that these models that were fictitious and hybrid is fictitious with 2 learning rates, were generally better than anything else at approximating behavior. And the only other models that were almost as good was when we introduced a decay of expected values. Remember, I've shown you that the probability of staying increased over time. We assumed that this was due to the animal could be forgetting what happened before. So we assumed that the expected values would decay over time and did an exponential decay of those values. And these models were sometimes better than the other, but this was not a general trend. However, they were only better when we paired with the fictitious updates, so still fictitious best and decay may be better. What is the big conclusion from this? Well, we have a model that probably, well, maybe let's go through the parameters first. So one important thing is that, remember the parameters in the Skinner box modeling? They were in the decimals, I mean 0.5, 0.6, 0.3. In this model with long time, free choice, the learning rates are extremely low. The positive learning rates are in the 0.01 range, and the negative learning is essentially 0. The beta values seem okay, both in this one which accounts, the basic one and the one which accounts for decay of the expected value. But it seems like they would only learn from positive outcomes and very little, and essentially nothing from the negative outcomes. And just for comparison, the Skinner box, these values were on a completely different range, although the beta values were comparable. Okay, so the conclusion from this is that we have a model that is able to approximate decision process under conditions where animals live in a group. They can perform choices whenever they want. They have 4 weeks to perform all the choices, and they are not forced in any way. They are not deprived from food. They are not deprived from water. They can just go for a drink of something sweet or something sweet with alcohol, and great. It's anyway anytime they want, but it's not required. And under those conditions, still the best we have is that fictitious update approximates animal behavior better than other approaches. We don't yet have an idea how the time effect should be accounted for, but we kind of see it in all our experiments. And if you have any suggestions, you are very welcome. And I'm finished, but I just wanted to say this was the work of many people, especially Teja Błońska, Przemysław Cieślak, Łukasz Szumiec from our side. And we really had, well, at the time we started, we had no idea about reinforcement learning. And Wu Yang An and Rafał Bogacz taught us everything. And the transgenic mice is work with David Engblom and Ginter Schutz. Thank you. I would like to thank all the collaborators and thank you for your attention.
Thank you very much for a fascinating lecture, and we are open for questions. Okay, please, gentlemen. Here.
Please wait for the microphone.
Thank you. Yeah, hi. Thank you so much for your very interesting presentation. My question is, do you think there might be a component of poor initiation or coordination of the movement and actually slowness of the movement itself in the selectively mutated NMDA receptors, and how that might be, I wouldn't say like a confounder, but maybe like an effect or an early effect in your experiment?
So you're absolutely right. We don't think they have slowness of movement because we tested them for Parkinson-like behavior, but since the dopamine system controls initiation of action, then if you look at this data I have shown you, this is the delay from the start when the queue is lit to the choice, right?
Yeah.
And okay, I say they are slower, so they're slower in making the decision. But here's the delay between the choice is made, and moving to the food port, and they are slower too. And there is no decision here. This should be automatic, and they are still slower. So you're probably right. These animals are probably impaired on how fast they initiate action. So I don't know, call it reflexes, I don't know.
Or even maybe due to particular areas in, like, I don't know, in the basal ganglia, say maybe for in caudate nucleus or something,
Well,
that might be attributed to this kind of effect?
Well, we looked, because theoretically, they don't have a deficit in dopamine. The tonic activity of dopamine neurons works perfectly well. Any local mechanism controlling release in the striatum work normally. The only thing is they don't have burst activity. I would assume that initiation of movement may require burst activity. It's certainly possible without it, but it's maybe somewhat slower. So they're not slower moving. I think that they are slower initiating.
Thank you.
Okay, Adam in the back.
Thank you very much. Great talk. Thank you. I get a question about the IntelliCage and the gambling task, because we tried to implement the Iowa gambling task in the IntelliCage, and there was a problem about copying the behavior of, in the group. So we made it in the water with the water, and we've got completely in 4 cages. We got completely 4 different strategies, group strategies. Depends on the where is the leader of the group, or and we check the followings and all those things. And how do you manage this effects of the group?
Okay, so obviously I should have stressed this more. These experiments are performed with 14 mice present. So there are 14 mice making the choice, so they are together. They can watch what the other mouse is doing, they can smell the other mouse where it's exiting the corner, etc. And we actually did try to make paradigms where the mice, when different mice had their wards in different corners, to kind of see if they will follow the other mouse or they will do the right thing. But these experiments are underway. There are colleagues of ours who are doing the research on this, and this is Daniel and also Michał here. And I'm afraid in this specific case, we could not observe any social effect. I mean, maybe there is, but we cannot find how to point that this specific behavior was influenced by the social interaction. And the Iowa gambling task is far more complicated than what we did. This is much simpler.
Any other questions? Okay, here. Sorry.
Hey, so regarding the, what was it? Shit. You were mentioning about potentially forgetting as an explanation of why people why mice over time stay instead of move around, and I was wondering, because there is this shift over time in the probability of of getting a reward, whether there's possibly a learning that what I've learned over time is no longer relevant, and so there's no need for me to move around. And how would that be tested even?
Okay, so when we were in the discussion for this, we were reading the literature on foraging. It's a foraging problem. When animals forage, they need to remember the previous location they were. And if they make a break, they often return to the last location they were, irrespective of the state of the area. But I'm taking this slide to show something. If it was forgetting, then I would expect this curve to be asymptotic to the 0.5. If it's forgetting, it should go to random choice. But it's not going to random choice. The longer time, the higher the probability. It really goes over 0.5 very significantly in most cases. So we use this forgetting approach because there is previous literature saying that forgetting occurs on scale of hours.
Yeah, that's what I'm saying. If it's not forgetting, couldn't it be learning that the expectation of choice is irrelevant after a certain period of time?
Well, that would be not model-free approach. The animal would have a specific system to say, okay, every 3 hours I should reset my expectations because the environment changes. Or it should have an idea that maybe every second day the conditions change and should be able to count days. And we assume it cannot count days. I mean, such assumptions require for the animal to have some model of the environment, which we assume it has not. Of course, we cannot know what the animal actually uses as the basis for the decision, but we assume it makes no such assumptions. That this is a semi-automatic process, model-free, as simple as possible.
Yeah, fair enough. Thanks.
Okay, one more question here.
Kind of follow-up question, because you mentioned the foraging and also assumptions of animals, but for example in free foraging, I think seabirds, where they basically don't have any cues, there's just endless sea, they still make these pretty interesting patterns in changing the area where they forage, and this might actually be hard-coded in kind of genetically hard-coded for optimal strategy of search of food. So can such a process be interacting with learning, basically, here?
It's very likely, but then what we want is to build models that predict animal behavior. And the question will be how to code it into the model. And for that we have no idea. Because what we want is a good model of how the system works. It's not important to us how good the model looks on paper. It has to be better at predicting the behavior, and it has to have as few parameters as possible.
Okay, thank you very much. And this is